Here are our top 5 stories for today, July 6, 2023. First story. Caribou Biosciences announces $25 million equity investment from Pfizer. Caribou Biosciences has announced a $25 million equity investment from Pfizer Inc., a global biopharmaceutical company. This investment involves Pfizer purchasing 4,690,431 common shares of Caribou at a price of $5.33 per share. As part of the investment, Sriram Krishnaswamy, Ph.D., from Pfizer's Global Product Development, has joined Caribou's Scientific Advisory Board. Caribou Biosciences plans to utilize the investment to advance their immune-cloaked allogeneic CAR T-cell therapy, CB-011. This therapy is currently being evaluated in the Camouflage Phase 1 clinical trial for patients with relapsed or refractory multiple myeloma. Caribou will retain full ownership and control of its pipeline of allogeneic CAR T and CAR NK cell therapies. The investment from Pfizer highlights the potential of Caribou's clinical programs and signifies a partnership with one of the world's premier biopharmaceutical companies. Caribou is actively progressing its allogeneic CAR T-cell therapy pipeline and will provide updates on various programs, including the Antler Phase 1 clinical trial for CB-010, the Camouflage Phase 1 clinical trial for CB-011, and the submission of an investigational new drug application for CB-012. CB-010 and CB-011 have been engineered using CRISPR genome editing technology to improve anti-tumor activity. CB-010, Caribou's lead product candidate, is being evaluated in patients with relapsed or refractory B-cell non-Hodgkin lymphoma. CB-011 is an allogeneic anti-BCMA CAR T-cell therapy being studied in patients with relapsed or refractory multiple myeloma. Both therapies have received regulatory designations, such as Fast Track and Orphan Drug, from the FDA. Caribou Biosciences aims to advance their mission of developing transformative therapies for devastating diseases through the next-generation CRISPR platform. Their focus is on off-the-shelf cell therapies for hematologic malignancies and solid tumors. The $25 million equity investment from Pfizer represents a significant milestone for Caribou Biosciences, providing resources to further their research and development efforts and potentially bring innovative therapies to patients in need. For our second story we have: the FDA converts novel Alzheimer's disease treatment to traditional approval. The FDA has converted the approval status of Leqembi, lecanemab IRMB, for Alzheimer's disease from accelerated to traditional approval. This decision comes after a confirmatory trial, Study 301, confirmed the drug's clinical benefit. Leqembi is the first amyloid beta-directed antibody to undergo this conversion process for Alzheimer's treatment. The drug works by reducing amyloid plaques in the brain, a defining feature of the disease. Alzheimer's disease affects over 6.5 million Americans, causing progressive memory loss and impaired daily functioning. While the exact causes are not fully understood, the disease is characterized by the formation of amyloid beta plaques and neurofibrillary tangles in the brain, leading to the loss of neurons and connections. Study 301, a Phase 3 clinical trial, enrolled 1,795 patients with Alzheimer's. Leqembi demonstrated a statistically significant and clinically meaningful reduction in cognitive decline compared to the placebo group. The drug's efficacy was evaluated using various endpoints, including the Clinical Dementia Rating Scale and the Alzheimer's Disease Assessment Scale. Common side effects of Leqembi include headaches, infusion-related reactions, and amyloid-related imaging abnormalities, ARIA. ARIA can result in temporary brain swelling and bleeding, occasionally causing severe complications. Patients with the ApoE4 allele have a higher risk of ARIA and should be tested before starting treatment. Leqembi's approval conversion represents a significant milestone in Alzheimer's treatment. It provides hope for patients and their families facing this devastating disease. The drug's approval was granted to Eisai Inc., and its use should be initiated in patients with mild cognitive impairment or mild dementia stage of the disease. Our next story is about MaxCyte and Lyell Immunopharma. MaxCyte signed strategic platform license with Lyell Immunopharma. MaxCyte, a cell engineering company, has signed a strategic platform license with Lyell Immunopharma, a clinical-stage T-cell reprogramming company. Under the agreement, Lyell Immunopharma gains non-exclusive clinical and commercial rights to use MaxCyte's flow electroporation technology and expert platform. In return, MaxCyte will receive platform licensing fees, clinical milestone payments, and sales-based payments. The collaboration aims to advance the development of T-cell product candidates targeting solid tumors. MaxCyte's expert instrument portfolio provides clinically validated electroporation technology for scalable cell engineering. With high transfection efficiency, scalability, and enhanced functionality, the expert platform supports the next generation of biological and cellular therapeutics. Doug Duafler, president and CEO of MaxCyte, expressed excitement about the collaboration and the potential to support Lyell Immunopharma in developing solid tumor treatments for patients with unmet needs. Each strategic partnership for MaxCyte generates pre-commercial milestone revenue and often includes sales-based payments. MaxCyte has been dedicated to cell engineering excellence for over 20 years, with a focus on maximizing the potential of cells to improve patients' lives. Their expert platform, based on flow electroporation technology, offers solutions for the rapidly expanding cell therapy market, covering the entire cell therapy sector from discovery and development to commercialization of next-generation cell-based medicines. By providing technology, technical support, and regulatory guidance, MaxCyte aims to empower its partners in transforming human health. For more information about MaxCyte, visit their website and follow them on Twitter and LinkedIn. In our next story, Arrowhead Pharmaceuticals reports inducement grants under NASDAQ listing Rule 5635, C4. Arrowhead Pharmaceuticals has made inducement grants to 56 new employees as part of their employment agreements. The grants were approved by the Compensation Committee of the company's Board of Directors under Rule 5635, C4, of the NASDAQ listing rules. These grants, which are separate from the company's stockholder-approved equity incentive plans, provide employees with up to 96,550 restricted stock units. The units will vest annually over a 4-year period. Arrowhead Pharmaceuticals specializes in developing medicines that target and silence disease-causing genes using RNA interference, RNI. Their therapies utilize a range of RNA chemistries and effective delivery methods to trigger the RNI mechanism, which efficiently and durably suppresses target genes. RNI is a natural cellular mechanism that inhibits the expression of specific genes, ultimately affecting the production of specific proteins. By harnessing this natural pathway of gene silencing, Arrowhead's RNI-based therapeutics aim to treat challenging diseases. The company's expert instrument portfolio represents the next generation of electroporation technology for scalable and complex cell engineering. These instruments facilitate high transfection efficiency, seamless scalability, and enhanced functionality, making them essential for advancing biological and cellular therapeutics. Arrowhead's strategic partnerships generate pre-commercial milestone revenue, with many agreements also including sales-based payments. Arrowhead Pharmaceuticals is committed to maximizing the potential of cells to improve patients' lives. With over 20 years of expertise, the company has developed best-in-class platforms and innovative solutions for cell engineering. Their expert platform, based on flow electroporation technology, supports the rapidly expanding cell therapy market. It can be utilized across various stages of the cell therapy sector, from discovery and development to the commercialization of next-generation, cell-based medicines. Our final story is about Aethlon Medical appoints medical device and biomedical executive, Nicolas Gikakis, to its board of directors. Aethlon Medical, a medical therapeutic company focusing on cancer treatment and life-threatening infectious diseases, has appointed Nicolas Gikakis to its board of directors. Gikakis, a medical device and biomedical industry executive with over 20 years of experience, will also serve as a member of the Nominating and Corporate Governance Committee. Aethlon Medical has increased the size of its board to accommodate the appointment. Dr. Charles J. Fisher Jr., CEO of Aethlon Medical, expressed his enthusiasm for Gikakis joining the board, noting his extensive experience and relationships in the industry. The company is committed to researching and clinically developing the Hemopurifier, a therapeutic blood filtration system capable of binding and removing harmful exosomes and life-threatening viruses from the blood. They are also exploring the potential utility of the Hemopurifier in the organ transplant market. Mr. Gikakis expressed his excitement about joining Aethlon Medical at this crucial stage, recognizing the vast potential of the Hemopurifier in oncology, the treatment of life-threatening diseases, and the organ transplant market. He looks forward to collaborating with the board and the Aethlon leadership team to drive long-term growth and value creation. Prior to joining Aethlon Medical, Mr. Gikakis held positions at Ware Optimo T Limited and Oventis Medical Limited, where he played significant roles in commercial expansion and corporate development. He also has a background in sales, marketing, product development, and corporate transactions in the healthcare industry, particularly in blood filtration and purification. Aethlon Medical focuses on developing the Hemopurifier, a clinical-stage immunotherapeutic device designed to combat cancer and life-threatening viral infections. The Hemopurifier has shown promise in removing life-threatening viruses and harmful exosomes from the blood, with potential applications in cancer and infectious diseases. It has received breakthrough device designations from the U.S. Food and Drug Administration, FDA, for the treatment of advanced or metastatic cancer and the treatment of life-threatening viruses without approved therapies.